Optimal health for high performers. This is the Health Upgrade Podcast with Dr. Nawaz Habib.
Hello, everybody. Welcome back to the Health Upgrade Podcast. I'm here with my co-host, JP. Great to see you, JP.
Great to see you.
So lately, I've been going down this rabbit hole of how vagus nerve is involved in nitric oxide. I believe that nitric oxide is such an important molecule within our bodies and has such a broad number of mechanisms that it's going to affect that the initial connection here came from this idea of how exactly within the cell is the acetylcholine from the vagus nerve affecting the changes. How does it allow for peristalsis in the gut? Or how does it involve and allow for movement of blood flow effectively through blood vessels? And that was really the starting point. And then I went down this rabbit hole, and it's been really quite wonderful and very insightful for me. So today's episode is going to be more of me kind of sharing some of the learnings that I've had, and for you to ask me a lot of questions, JP, as we go through. All right?
Sure. You certainly have taken the deep dive into this subject. I would love to ask you a couple of questions right off the bat just to get started. So nitric oxide can be generated in the body in a variety of different ways. Why don't you tell us about the different pathways to getting to nitric oxide generation?
There's two particular big pathways by which we produce nitric oxide within the body. One is a dietary requiring pathway, and that is called the nitrite nitric oxide synthase pathway, or nitric oxide pathway. Remove synthase from that. It's literally a requirement to bring in nitrate from dietary sources. And these include foods that are high in this potassium nitrate in particular. So things like beets, things like arugula. These are foods that primarily are plant-based that are going to provide a lot of these nitrates, inorganic nitrates, into the diet. And these nitrates are actually converted to nitrite within the oral cavity by the oral microbiome. And this is a really important thing that I think has been overlooked for a long time. The oral microbiome is part of the gut microbiome, as the mouth is a part of the gut. And the specific probiotic or the specific bacteria that live within the oral cavity are required to convert that nitrate to nitrite within the oral cavity. And then that the nitrite, this is going to be a lot of nitrite stuff. So we're going to have a lot of these tongue twisters going on. But the nitrites that we've had converted by these oral microbiome bacteria actually then store the nitrites within the saliva, in the salivary glands and within the oral cavity. And every time we require release of these nitrites, it comes out within our saliva, and we're then able to convert nitrite to nitric oxide lower down within the gut and within other tissues of the body. And so that's one pathway by which we can get the nitric oxide that our body requires through dietary sources in particular.
And in combination with the microbiome in the mouth and in the gut.
Exactly. So gut microbiome and the particular bacteria that live within the microbiome are of very high importance when it comes to that pathway. So that's a really important one to look at.
Okay. Now, but there's other ones.
Yes, there is a second pathway. And this is the one that we're all blessed with at birth. And we have this genetic pathway internally within us, which is the nitric oxide synthase pathway. And this is the actual use of an enzyme that's present in all of our cells, but in particular cells, there's different forms of it, called nitric oxide synthase. There's 3 different forms of nitric oxide synthase that all work to convert a very important amino acid, L-arginine, into nitric oxide, and we come out with a separate amino acid called L-citrulline as the byproduct of this. So we take L-arginine and we break it down into nitric oxide and L-citrulline, and the nitric oxide is then utilized by the cells for whatever the function is in a regulatory fashion or in an inducible inflammatory fashion, which we'll get into. So there's that specific pathway, the nitric oxide synthase pathway, is the one that we have genetically within our own genome, within our own enzymatic structure in our own cells. So there's the two sides to this coin.
And there's three forms of that nitric oxide synthase.
That's exactly right.
And one of them you hinted at was associated with inflammation.
Yes.
I want to spend a minute or two talking about that.
Yeah. So there's three isoforms here. The one that we're talking about when it comes to inflammation is one called iNOS, the letter I, which stands for inducible nitric oxide synthase. So inducible nitric.
It's the Apple version. It's the iNOS.
Exactly. That's funny. I didn't even think about that. But yes, it is the Apple version, quote unquote. It is the one that is involved in the inflammatory process. And so the cells that tend to have inducible nitric oxide synthase, that isoform, active, tend to be heavily inflammatory cells. Things like activated microglia, activated macrophages. These are the M1 type macrophages. These are the M1 or the macrophages that are in their fight state. They tend to be stimulated more by either sympathetic activation or threat response. And so if there's a threat that's there, something like a cancer cell, for example, or a bacteria that's present that needs to be devoured and phagocytized, it's the macrophage that produces a high level of nitric oxide through that iNOS pathway that allows for relaxation of the form of the cell to then go and engulf the threat to our survival. And so it'll go and break it down accordingly.
So this nitric oxide release or generation is used by inflammatory cells to promote or facilitate their motility, their movement.
That's exactly right.
Now, is that nitric oxide released internal to the cell or external or both?
It's both. So it's going to be internal to allow for the formation of the cell to shift, and then externally to add a ton of free radicals to try to kill off the surrounding threat or whatever that threat is in its surrounding.
And so this iNOS pathway, this nitric oxide synthase is the I, assumingly, is not an Apple thing. It's an inflammation immune. But is that enzymatic reaction taking place in the vicinity, inside the cell, in the cytosol, or is it in membrane bound, or where is it located?
Most of the time, it is a membrane-bound nitric oxide synthase isoform. So it's sitting on the surface of the cell, and so it will have the ability to send nitric oxide in either direction, either extracellular or intracellularly, to allow for that motion to occur. The really important thing to note with iNOS itself is that it produces a very, very, very high amount of nitric oxide. And it does not require a calcium channel to be activated to allow for that pathway to occur. So in order for iNOS to have its effect, it requires arginine, which is a non-essential amino acid, meaning our body can produce arginine on its own, but it does not require that calcium to have a regulatory effect. It's essentially producing this nitric oxide to devour and kill off the threat to our survival, and is occurring in a very, very high quantity. When it does so, because it's doing this without that calcium-dependent pathway, it has a high amount of reactive oxygen species that it's going to produce. And in an acute circumstance, that's fine. But in a chronic circumstance, a high level of reactive oxygen species is going to have an effect on our aging. It's going to kill off good cells. It's going to have an effect on DNA in other cells as well. So this is a very important piece to the puzzle on how chronic inflammation leads to breakdown of other cells in the area. So it's the control of that inflammation that needs to occur. And in this particular case, the way that the vagus nerve controls the inflammation within the macrophage is by downregulating iNOS activity.
Well, and this is really important for, and we're going to get into this, I know, that release of nitric oxide external to the cell has the ability to relax smooth muscle. And so we're going to talk about the cardiac effects. But first, just getting back to the roots that I have in vagus nerve stimulation, which is in migraine, the effect of the release of nitric oxide by these inflamed macrophages can lead to a relaxation or an expansion or dilation of the cerebral blood vessels. And as a result, you get extravasation of fluids and other cells into the brain, which can cause pain. And so one of the pathways that's believed to be associated with migraine is this INOS pathway, which is releasing nitric oxide and leads to this extravasation of fluid into the brain, causing pain. That's why people who have migraines feel it's a pulsing pain that's sort of synchronous with your blood pressure or your heart rate, because you're actually causing those blood vessels to release more and more of this pain activating volume, if you will, as a result of this. So blocking INOS through blocking inflammation has an effect on migraine.
100%. And it's that cortical spreading depression that is being downregulated when the vagus nerve is being stimulated and the acetylcholine is being released effectively. And so that's what we're looking at for the regulatory or to downregulate that INOS activity. But yes, you're absolutely right. If we're creating a vast amount of nitric oxide within the entire vasculature of the brain, then what's happening is these blood vessels are literally growing into a larger size. They're dilating as much as they can. And that dilation leads to pressure being built around the blood vessel in the areas around the brain, which is why we then feel almost like a pulsatile pain feeling with migraine. We can almost hear our heartbeat in our brain. And that's how migraine works. That's literally how that mechanism occurs, is due to the high level of INOS activity when these microglial cells are in that heavily microglial activated M1 phase.
Now, the interesting thing is that with migraineurs differ from what we'll call healthy, normal people who don't suffer with migraines, in that if you give a person who is a migraineur an NO donor, because nitric oxide is NO, if you give them an NO donor, like for example, nitrous pills that they take, almost like TNT, actually, which is good for your heart. It relaxes the heart muscle. And we're going to get deep into that. But it actually gives people who are migraineurs a migraine. It can trigger a migraine. Whereas for a person who doesn't have migraines, they might experience a mild headache for 10 or 15 minutes, but it goes away. But you can get pushed into a full-blown headache, full-blown migraine as a result of this.
And that's exactly why it seems like the mechanism by which migraine is going to occur is not necessarily like the nitric oxide is the pathway to create the pain, but it may actually just be a lack of acetylcholine that's allowing for the regulatory situation to occur within the immune system of the brain, within the microglia itself.
Well, since we've hit on migraine and how vagus nerve stimulation can treat migraine and how that entire pathway involves the release of acetylcholine, et cetera, let's dive into the other two ways that your body synthesizes nitric oxide in the cells.
Yeah. And so there's the two other isoforms of nitric oxide synthase that are actually really important and really regulatory. And this is really important to understand. So we've got the INOS, which we talked about. That's the inflammatory inducible nitric oxide synthase, and that's involved in the inflammatory pathway. On the regulatory side, we've got two isoforms. One is called endothelial nitric oxide synthase, eNOS, and one is called neuronal nitric oxide synthase, and that's the neuronal version, and that's involved in nerves being able to send nitric oxide in smaller doses to certain areas to allow for relaxation to occur. So nitric oxide can have both a really good and positive effect and a really, really negative effect, depending on how much regulation there is occurring and the type of, or the isoform that's being utilized to produce the nitric oxide to begin with.
Let's talk for a second about the nNOS.
Yes.
So how is the nitric oxide that's generated by the neuronal nitric oxide synthetase, how is that released? Is it released synaptically? Is it released extrasynaptically? Does it remain inside the cell? What's its function?
So these ones are located, the neuronal nitric oxide synthase is located on the extracellular surface, on the membrane within the synapse. So it is releasing nitric oxide into the synapse of the cell. So where the nerve attaches to whatever the the tissue is in its region, wherever that attachment is occurring, the nitric oxide is going to go to that postsynaptic membrane. That postsynaptic membrane can be the heart, it can be in the gut, it can be along the lines of any tissue where that nerve is attaching to. And it's going to have a very specific regulatory effect.
So within the brain, you've got neurons synapsing with other neurons, and you've got astrocytes, which are regulating the volume, if you will, controlling the extraction and the rate of neurotransmitter flow through that synapse. Does the nitric oxide affect the astrocyte, or is it only affecting the postsynaptic terminal?
It wouldn't surprise me if there was an effect on the astrocyte itself as well. From my understanding, and this is my hypothesis here, is that nitric oxide is the great relaxer. And what it's going to do is it's going to allow for motion in conjunction or in almost a pulsed form with acetylcholine. So acetylcholine is the great contractor. So it's what's involved in the contraction of muscle. It's the signal that's required from the acetylcholine that is then released on smooth muscle to create a contraction of the smooth muscle in the gut, in the blood vessels, etc. And it needs to almost be pulsed with nitric oxide, which is a great relaxer. So we have a contraction, relaxation, contraction, relaxation, acetylcholine creating contraction, nitric oxide creating relaxation. When there's too much relaxation, when there's too much nitric oxide, we can get into a situation where there's a hyper relaxation effect, and that cortical spreading depression can occur within the central nervous system. That's also going to occur when we have almost like in the heart, a bradycardia. So we go into a too slow and too relaxed heart situation, and that's not good either. We need to have a back and forth and a almost one-to-one type of response between acetylcholine and nitric oxide to occur. And so that one-to-one response tends not to occur with iNOS. iNOS is nitric oxide first, very little acetylcholine, versus eNOS and nNOS, which are both kind of that one-to-one nitric oxide and acetylcholine allowance. And so we almost get that pulsed effect that's going to occur. It'll happen in a slightly different way between the endothelial and the neuronal version, but those two isoforms are much more regulatory, and it's actually acetylcholine that's driving the function of eNOS and nNOS, depending on where they're at.
Well, I know we're going to get into a couple of papers that talk about how vagus nerve stimulation can help to regulate that release of nitric oxide in the context of heart failure and in the context of cardiovascular problems. Let's talk about the fact that because the cardiovascular system is highly innervated. Obviously, your autonomic nervous system is controlling both your heart rate as well as the tension or the level of tonicity in the vasculature itself. But there's endothelial cells that are part of that equation. And if there's the eNOS, what portion of the control of your blood volume, which is controlled by the level of tonicity in the blood vessels of your vasculature, how much of that is eNOS generated versus nNOS generated? Because I would imagine a little goes a long way with nNOS. A little goes a long way, and it probably triggers the eNOS. So let's talk a little bit about how the interplay between them.
Yeah, so it depends on the location of the blood vessel that's going to have that different effect. So certain blood vessels that are highly innervated, things like the aorta, the aortic arch, the carotid artery, they have significant amount of innervation. And so they need to have a good amount of that innervation because they require more of that pulsatile relaxation contraction to be able to maintain blood pressure. And so that's a really important interplay between the neuronal side as well as the endothelial side. And I believe that there's a reason why this occurs, and that is that where the blood flow is significant in those major arteries, where the blood flow is really fast, where there's high pressure that's occurring, there isn't enough time for the endothelium or the endothelial cells to receive acetylcholine from the blood cells that are present in there as well. I believe it has to come from a neuronal source in order to allow for that relaxation to occur. And like you said, a little will go a long way on that neuronal side. And it's synaptically located, right? Like it's on that synapse. So it's showing up on the blood vessel side, on the smooth muscle very quickly from a neuronal side, rather than needing to require the chat cells to send acetylcholine from the inside of the blood vessel. And there's not a lot of time there.
I was going to ask you that because you mentioned just a minute ago, you said, there's not enough time to get the acetylcholine to the endothelial cells in those large and really important blood vessels that have to respond immediately to the contractile pressure of the heart. So, but I was going to ask, it's obviously not just acetylcholine floating through the bloodstream. It's delivered to those cells or that tissue by cells that are floating through the bloodstream, and those are the chat cells. Those are your T cells that are able to release acetylcholine. And they're really important cells in various aspects of vagus nerve stimulation. We've talked about them in the context of the spleen. We've talked about them in the context of the gut. What role are they playing in the smaller blood vessels? Because of course, yes, it's really important to control the aorta and your carotid artery, et cetera. But peripheral vasculature and even the microvessels, it's really important to maintain the tonicity because you can't get blood through them if the tone isn't right.
That's exactly right. And we're not going to have nerve endings on capillaries, right? We're not going to have nerve endings on the smaller blood vessels. They're just too small to allow for that extensive a neuronal network to exist. And so these T cells that have been activated through the vagus nerve via the celiac ganglions, the splenic nerve to the spleen, activating those through the beta-2 adrenergic receptors on the chat cells, those T cells, those CD4 plus lymphocytes go out and they release acetylcholine via the bloodstream. And in the context of inflammation, that's how we get the acetylcholine to the tissue-resident macrophages and cells that are not directly innervated by the vagus nerve. But those chat cells are also producing that acetylcholine while they're flowing through the bloodstream, through those smaller blood vessels. And so it's going to release acetylcholine in those smaller blood vessels, and that acetylcholine is going to attach to the muscarinic acetylcholine receptors on the surface of these endothelial cells, the lining cells of the blood vessel. That acetylcholine is then going to trigger the nitric oxide synthase eNOS activation within the endothelial cell, and that eNOS is going to produce nitric oxide through the L-arginine pathway and send that nitric oxide to the smooth muscle cells one level below the endothelial cell, which is then going to allow for relaxation of the blood vessel to occur. It's going to allow for that through the CGMP pathway, relaxation of smooth muscle to occur.
So we didn't talk about this beforehand. So I'm going to hit you with something that we haven't prepared for at all. But there's a condition called Raynaud's syndrome.
Yes.
And Raynaud's is a situation where people will move into, for example, into cold water or a cold room that's highly air conditioned, et cetera, and all the blood flow to their fingers will just stop.
Yes.
It's been suggested that vagus nerve stimulation may be useful in treating that, which suggests that there's an acetylcholine aspect to this, which of course, if you're talking about the blood vessels being too contracted to pass blood flow through it, then you would expect that something either like eNOS or nNOS, probably more nNOS, is at play here. Any thoughts on this? Does that make sense to you?
It makes a ton of sense to me, and it really has to do with the contrast between those nitric oxide and ACH pulses that almost need to occur to allow for that rigidity not to occur. So this is for anybody who says like, man, my circulation is poor, my fingers are always cold, my toes are always cold. And then it can go into a full disease state where you actually lack blood flow into those areas. And that I absolutely feel has a very important role to play. And that's where we often are lacking not only the vagus nerve function and the vagus nerve tonality to begin with, but now we're also then unable to produce the acetylcholine effectively through chat cells. And we're likely lacking from a nutritional standpoint the components that build up acetylcholine to begin with. So 90% of diets are actually deficient in choline which is a really important piece to the puzzle. And mitochondria are required to produce the acetyl-CoA that's going to be the paired piece to the acetylcholine. And so if we lack mitochondrial function and we lack choline, then we have a reduced capacity to build acetylcholine to begin with. And that's where the potential mechanism is to producing this condition of Raynaud's.
Well, Raynaud's is a really important condition, especially for those people who have it. I mean, people can lose digits. It can be debilitating. But let's get into talking about the cardiovascular, the severe cardiovascular conditions where producing enough nitric oxide is critical for life and survival. We found a couple of papers, one from back in 2009 and one more recent in 2022, that talks about how vagus nerve stimulation can be used in the context of heart failure and fibrillation to enhance the health of the patient, both in the short term, preventing sudden onset of death, as well as just the overall health of the patients. Which one would you like to tackle first?
Let's start with the 2009 one.
Makes sense. What would you like to say about it? It's a great paper, and I think in some respects they did the experiment better back then in 2009. But let's dive into it.
Yeah. So essentially, what they did was they tested different frequencies of stimulation of the vagus nerve and checked to see what was going on with regards to the heart itself, and if there was actual changes in nitric oxide synthase activation, neuronal NOS activation within the left ventricle of the subjects. And what they found was there was a dose-dependent and frequency-dependent response. So what they showed essentially was left and right, not a huge difference in terms of which vagus nerve was stimulated. We're going to have much of an effect, but the higher the frequency, the more nitric oxide was being produced through that neuronal nitric oxide synthase pathway at the ventricle. So this was a really important finding.
So it's interesting that you said they didn't find very much difference between the right side and the left side, because a lot of the original work done in vagus nerve stimulation for epilepsy, they focused on implanting the devices on the left side. And the reason was they felt that somehow that was going to provide less of an impact on the heart, because they were worried that they were actually going to have negative impact. We're now realizing that no, it was actually a positive impact on the heart. But what you're saying is that whether you do the right or left side didn't matter very much with respect to the effect, at least in this effect, on the heart tissue and the release of nitric oxide. Interesting. Now, when you said that they used different frequencies in this 2009 study, they were studying 5 hertz, 10 hertz, and 15 hertz.
That's correct.
None of which are frequencies that are currently used for therapeutic purposes. We're typically usually above 20 at 25 or even 30 hertz. Some people are looking at uses up at 40 hertz. Was the trend towards better NO production the higher the frequency?
That's exactly right. So they found increased, and obviously based on the study parameters, they only got up to the 15 hertz mark, but they did see a very significant difference between the 5, the 10, and the 15, and the 15 was the highest by a large margin.
Excellent.
Yeah.
So what other things should we draw from the 2009 paper before we jump to the 22 paper?
I think it was important to understand that it's an acetylcholine dependent function that allows for the nitric oxide synthase activation to occur. So when the vagus nerve signals, it doesn't signal nitric oxide itself. It signals acetylcholine to the postsynaptic interneurons that are present within the heart. And so in the left ventricle, where we're getting that large contraction that's causing the blood to flow through the entirety of the body, basically, we require a lot of relaxation initially and a lot of pulsing with muscular contraction. And so that requires a very rapid amount of nitric oxide to be released to allow for the ventricle to relax, and then a significant acetylcholine pulse to pulse and contract the heart, the ventricle itself, to allow for that ejection of blood into the aorta. So for me, that was probably the biggest thing, showing that it is an ACH dependent neuronal nitric oxide synthase activation that's occurring, and then it happens very rapidly in milliseconds to allow for this to occur.
So it really has to be neuronal, because anything going through the circulation would just wouldn't be fast enough.
Yeah.
So it's got to be neuronal.
Funny enough, that neuronal pathway is the same one that's required in peristalsis and the migrating mortal complex in the gut. As a bolus of food comes in, the stomach needs to relax and allow for the food to enter. And then it needs to contract to push that food, that bolus, further on throughout the entirety of the digestive tract. And that has to occur not only in the stomach, but at every sphincter. It has to occur throughout the entirety of the length of the intestinal tract, small and large. That whole thing is a pulsed response, with nitric oxide allowing for the relaxation and acetylcholine allowing for the contraction behind the bolus to literally push the bolus along the tract.
And there's obviously a role for serotonin in peristalsis. That's where it was originally discovered. And so how does serotonin play into that peristaltic motion as well? Because what you're saying is all controlled. The actual flexion and relaxation is associated with nitric oxide and acetylcholine, but the controlling of that is one level higher with serotonin.
Exactly. It's the serotonin that is affecting the sensory nerve at the area where the bolus is entering. So the serotonin and the stretch signals are coming in to allow for the stretching of the intestinal tract to occur. And that is then signaling to the interneurons of the enteric nervous system to produce either nitric oxide in the area where the relaxation needs to occur, or acetylcholine to the area where the contraction needs to occur. And serotonin is heavily involved in that pathway.
So it seems like a pretty easy feedback to understand, that the stretching leads to the production of nitric oxide so that there can be relaxation, but the nitric oxide triggers the serotonin level to release the acetylcholine to then come in behind it and cause the contraction. Interesting. And that's in the gut. Similar things going on in the circulatory system. I mean, I love how Mother Nature uses the same things in multiple places where she needs it. In the gut, maybe it's a little slower, but you need to sort of push things through the system. Your vasculature, we have the heart, obviously, is doing most of the pumping, but the vasculature itself has to respond to that and contribute a little bit. We talk about the contribution of the vasculature to the overall pumping mechanisms of the circulatory system. So maybe we switch to the 22 paper and talk about what they did there.
Yeah, absolutely. Why don't you dig into that one briefly right off the top?
Yeah, so I actually found it sort of, I won't call it disappointing, I'll just say surprising to me, that they didn't look primarily at higher frequencies. A lot of what they did was, and they were using rabbit hearts, and they were looking at lower frequencies, 1, 2, and 3. They saw the same thing again, where higher frequencies, even within that small range, seemed to produce a stronger NO effect. They did do some work at 5, 10, and 20 hertz, but that didn't seem to be the focus of the paper. Again, finding similar results, that vagus nerve stimulation caused a significant release of nitric oxide in the left ventricle to drive the pumping action of the heart, and that vagus nerve stimulation could be useful even in the absence of change of heart rate. Yeah. So a lot of the work that's been done in vagus nerve stimulation has had as a concern the bradycardia that you talked about, the lowering of heart rate. There's usually a modest lowering of the heart rate, but that modest level is really associated with freeing up the flow of the blood through the system. So it actually decreases the strain on the heart to fight against the pressure in the system. But
I think it comes down to efficiency, right? It makes the system far more efficient and doesn't require the heart rate to drop off, but it drops to a good regulatory level, a level that is ideal for efficiency.
Right, exactly. But if you stimulate a different set of fibers in the vagus nerve, which are the C fibers, these are the unmyelinated C fibers, they tend to they tend to have a very profound drop in heart rate, triggering bradycardia. They also simultaneously have the other vasovagal responses of syncope, you can pass out, and bronchoconstriction, bronchospasm. So we talked also, maybe it was off camera, we talked a little bit about stretch receptors in the lungs and how that same stretch response that leads to the production of nitric oxide is useful in the lungs, because as you expand your lungs, as you're trying to take a deep breath, which is difficult for asthma patients, which is another place where vagus nerve stimulation is very beneficial. But when you're trying to take that deep breath, you're fighting against the musculature that's in that surrounds the lungs. So what you want is a release of that nitric oxide to allow your lungs to expand more, and inflate more and be able to take in oxygen, and then have that that subsequent release of acetylcholine to allow it to compress and you can breathe out. So when that cycle is not functioning properly, you end up with asthma. When you're not functioning properly, you end up with heart problems simultaneously in the cardiovascular system. And so I think this paper was interesting. It sort of followed on that work, but I was personally taken by the fact that they didn't spend as much time focusing on the higher frequencies, which is where I think the beneficial, the therapeutic benefit will come. And they discussed the fact that there were early papers that showed benefit in using vagus nerve stimulation for heart failure patients, but that they got sort of mixed results later studies. And I think that may be a function of the change of parameters, and maybe it needed to stay at the higher frequencies and not go to lower frequencies.
Which makes a lot of sense, right? And that's what we're seeing is it needs to, these higher frequencies tend to have a much more therapeutic benefit. And so that's ideally where this research goes over the next little bit, to prove that the higher frequencies are what's required to enable that pulsatile vagus nerve acetylcholine release with that pulsed nitric oxide synthase activation. And ideally, that's how we can help support the health of many people that are dealing with so many different conditions that are involved in that lack of pulsatile signaling that needs to occur.
That's an interesting point, because we've talked now about the brain and the nervous system centered on migraines. We've talked about in the periphery, maybe peripheral vascular conditions like Raynaud's disorder. We've talked about the lungs and breathing and asthma. We've talked about the heart and heart failure, and then in the gut, we talked about that as well. Are there other areas where you think that vagus nerve stimulation and this triggering of nitric oxide might actually end up being beneficial, where nitric oxide is really important?
I think through the rabbit hole, I've realized that it's wherever smooth muscle is located and it doesn't have that pulsatile effect, we have rigidity that's going to occur, and that's going to result in a lack of good blood flow. It's going to result in a lack of good neuronal control, and it's going to essentially result in rigidity, aging, and early or premature death, which is really unfortunate. So I think it's an across the board, wherever there's smooth muscle, that's where we really need to look at, is there enough acetylcholine coming in to signal for the production of both acetylcholine release for contraction and nitric oxide for relaxation?
Is there a relationship? I mean, I know that they discovered a lot of the ED drugs as a result of looking at cardiovascular effects, and it didn't have a strong enough effect in cardiovascular to end up, for example, Viagra becoming a drug in the cardiovascular space, although it is still used off-label for that, actually. Yes. But it obviously had this effect on erectile dysfunction.
Yeah.
Do you think that vagus nerve stimulation and the release of nitric oxide that's triggered through the nervous system and through the endothelial tissue could end up being having a role there? I mean, there hasn't been a lot of data.
It's certainly possible, but I think what actually is occurring is nitric oxide synthase as an enzyme is very, very sensitive to toxic burden. And as the toxins build up, as the challenges and the stressors build up, nitric oxide synthase actually becomes far less effective in producing that nitric oxide. And so what we can do over time, ideally, is to build that resilience and avoid as many of those toxins as possible. That's likely one of the reasons why it's leading to as much erectile dysfunction and challenge that people have been having. Where there's a lack of good blood flow, we're going to have a lack of nitric oxide function effectively. And so I think there's other factors that are going to be involved in nitric oxide synthase not being effective. And this is a huge sign, right? Erectile dysfunction is one of the first signs of this pathway not working. It's one of the first ones that pops up as people are aging, as they get into their 40s and 50s. They tend to, if they've been under a significant toxic burden or toxic load, and they have challenges like metabolic dysfunction or significant oxidative stress within their bodies, that is going to have a negative effect on the nitric oxide synthase in their blood vessels. And because the blood vessels within the penis and the erectile tissues tend to be quite small, those tend to be the first ones that are affected. They don't tend to have as much of the neuronal. They have a lot more of the endothelial nitric oxide synthase. And so there's probably a reason why they're the first to be affected in that way.
Interesting. Now, the circulatory system, blood flow, et cetera, is very important. But there's also the flow of lymph through lymph nodes, et cetera. Is there a role for nitric oxide in the lymph? I mean, it's something that I haven't seen many papers looking at it, but do you have any insights?
I think less so. And I believe it has to do with the fact that the main driver of lymph flow is actually muscular contraction. It's almost reliant on the muscles to be the pumps that allow for the fluid to move through the lymph. That there's not so much of an acetylcholine and nitric oxide pumping that occurs in lymph nodes and within the lymph kind of pathways that need to bring the fluid back up. So that has a lot more to do with exercise, muscle quality, muscle function, and movement, less so on the acetylcholine specific side. That said, acetylcholine is the main driver of muscular contraction through the other nerves, not the vagus nerve, but through the motor neurons. So that'll obviously play an important role there, but it's not a direct acetylcholine creating the pump effect.
Well, I know that we wanted to keep this relatively tight. As a parting discussion point, you brought up the fact that arugula and beets are good sources for nitric oxide precursors. Is there anything else that you would suggest from a dietary standpoint? And obviously probiotics, making sure that you have the right microbiome functioning. Yes. Anything else that you would suggest for people?
Yeah, oral and gut microbiome is huge. We want to also ensure that we have enough of the arginine. It is non-essential. That said, a good protein intake on a daily basis is going to help to ensure that we have enough protein present within the body to be able to pull the arginine when we need to. So a protein, wouldn't say heavy, but a protein efficient diet is going to be very beneficial. And we also want to look at the precursors of acetylcholine. And so B vitamins being a major piece to that puzzle, and choline, which I mentioned is quite deficient. Egg yolks are a great way to get this in. Sunflower lecithin is a great way to get this in. We're looking basically at a whole foods, healthy protein containing diet as being the main driver that allows for us to have good microbiome function, have good acetylcholine nitric oxide balance, and to allow for us to be able to build up the requirements, and then to do the vagus nerve activating exercises and potentially stimulate where need be to allow for that entire function to occur.
Agreed. Well, this has been very informative.
I love it. Yeah, this is good. This is my recent rabbit hole, and I've learned a ton, and I'm happy and excited to share this with as many people as I can.
Well, you did a great job of it, and I learned a lot just listening in. And thank you for forwarding the articles; they were really interesting. I know we're going to post those for the listeners to let them dig into those as well. And for everybody out there who's listening, if you've got somebody that you know and care about, or if you are experiencing issues with whether they be cardiovascular or respiratory, or if you have troubles with migraines, consider vagus nerve stimulation for yet another reason, because of its ability to regulate nitric oxide and acetylcholine.
Love it. Thanks so much. And we'll catch you guys on the next one.